Hello everyone, good afternoon, good evening, and good night to all. I am here to talk about the Laboratory 8 exercise. Now, for those of you who have gone through the laboratory exercise handout, you'll see that it has an introduction here present, kind of explaining what the entire exercise is about and the formulas that you're going to be using for this exercise. Following that is the first table and first set of instructions in this lab. Note that you do not need to submit these exercises. However, you are required to apply the knowledge to complete Lab 8. Here's the first table, and this table will serve as our dataset. As you can see, we have Community A and Community B, both of which have their own respective species that were found in the locations, as well as the number of species that were found in each location. At the bottom, there is the total, which you will calculate by adding up all of the numbers of individuals of each community and placing it in that box right next to the total name. Following this are your first set of instructions, and they are to define species richness, evenness, and diversity, calculate the species richness for both communities A and B, identify which community is richer than the other, and calculate the species diversity for both communities using equations 2 and 3. For that, we can use the following tables, 2 and 3, to help us identify them. Table 2 is dealing specifically with community A. You can set this up on the same Excel sheet or same Word document, however you want, but I suggest you split them up into two different tables so you have a better understanding of what's going on for each community. For each table, you're going to have different columns. The first column being the species, or in this case identified as I, meaning individuals, the number of individuals, denoted by NI, the proportion of individuals, denoted by PI, the natural log of the proportion of individuals, which is LNPI, and the proportion of the natural log of proportion of individuals. Yes, it's a mouthful, but don't worry, we're going to get to that. Now, each of these columns have their own respective formulas. If you're going to use any kind of system or software to assist you in this, I would suggest Excel, because you are able to write your own code or formula into the cells and then copy them to follow whatever information you need. For the NI category, this represents the number of individuals for each species. Now, for the key and I view, you'll notice that the number of individuals is actually a column under each section, Community A and Community B, in Table 1. That is essentially what you're going to be putting back here into this column, or NI column. For each Kiskadee, you're going to type out the number of individuals. So the Kiskadees are 69, so you type 69 under the first column, or into the first row of the column. For parrots, it's 35. For the Scarlet Ibis, it's 16. Hawks is 10, and so on and so forth. Following this is the proportion of individuals. In this case, this refers to the number of individuals divided by the total amount of species individuals present within the community. So, like before, when you found out the total number of individuals present within community A or B, this is where you'll see what percentage of a Kiskadee, for example, is present within community A or B. Following the proportion of individuals for each species, you're going to be looking at the natural log of the proportion of individuals, or percentage of individuals. For the proportion or percentage aspect in Excel, if you're using that software, type in the cells equals and also the cell with the number of individuals also divided by the total number of individuals. In this case, you can type it manually, or you can type the cell with the community A total. Once that's complete for each species, you can move on to the natural log of the proportion. This is also another simple aspect. You just have a calculator on standby, use the natural log function alongside the PI value that you have there, or in Excel, you can do the same thing where using the natural log function, and it does it for you automatically. Yippee! Now, for the final aspect of this table for community A, we're going to be doing the proportion of individuals multiplied by the natural log of proportion of individuals. Again, this is another simple formula which involves just multiplying the value for PI and the value of the natural log of PI. This can be done in Excel by doing an equal sign and then writing the formula outwards using the cells, or by typing it out manually. However, by using the cells, you're able to copy and paste down each line, allowing it for much easier access and convenience. For those of you who are doing this manually with calculators and not with computers or Excel, using a calculator is completely fine and you're completely okay for using one. For Table 3, you're going to follow the same process as you did for Table 2, but this time with Community B. So for all the members in Kiskadees for Community B, you're going to write down that in the NI section, and you're going to follow the same procedures for each of the following columns. Again, for those of you who are keen-eyed, you've spotted this much earlier in the introduction section of this handout, where it shows equation 3, the Shannon Diversity Index. Now, the Shannon Diversity Index, all those fancy symbols, they just mean one thing really. It means the sum of pi ln pi. So that means the total Shannon Diversity Index for Community A would be the sum of all the values for each species in the final column of each table, or under the pi ln pi section. That is why at the bottom of each of these tables we have a little total section, where you can type in the total of the pi ln pi. And this is where you get your Shannon Diversity Index. It is important to take note of all the elements in the formula for the Shannon Diversity Index. At the beginning, there is a negative sign, which is applied to the entire formula. This means that the ending, or the sum of pi ln pi, has a negative sign attached to it. Thus, any algebraic rules follow this procedure as well. The big fancy E that looks like a 3 backwards is known as the sum of symbol, and the S at the top refers to the species. I is the individuals, and it's set to 1 in this case. Or you could think of this as the species or number of individuals at base is 1. And the following expression, pi ln pi, as stated before, is simply the final part or the final value you get after completing each table. With all that added up and the minus sign attached to it, you get your final Shannon Diversity Index for each community. Now, this could mean very different things. For your lab report, you are going to need to understand why the Shannon Diversity Index is the value it is. Explain kind of why or what the value means in relation to each community. Furthermore, it's also important to think about why the Shannon Diversity Index is used and why either community A or B has a higher species diversity than the other. It is possible as well to be uncertain of when you have collected enough data or enough sampling has been done so that you can move on to data analysis or the measurement of the species richness, diversity, or any other determining factor. A good technique you can use in this case is a cumulative species sample curve. To perform this curve, we're going to be using Table 4, the survey of marine fishes, and we're going to be using this in a different way than the previous tables. Here we have all the species that were caught under each sample or sampling session, and then we have two other columns, the new species that were caught as well as the cumulative species. The way this section works is that for every new species present in the sampling sessions, for example, for sample 1, there was the gilbaca and the gray snapper, thus meaning that there were 2 new species caught from this session, also meaning that the cumulative species increases by 2. Following this, in the next sampling session, we found that there were gilbaca, gray snapper, red snapper, and bangameri. That means that the new species present, our red snappers and our bangameris, are added to the new species caught column. So there's 2 new species. There being 2 new species means that our cumulative species increases by another 2. In sampling number 3, there is the gray snapper, red snapper, bangameri, butterfish, cuffum, and high water, meaning that there's 3 new species, hence adding 3 to the new species caught column, and adding 3 to the cumulative species that is present. This process continues for each of the sampling sessions, for each new species caught, and each increase in cumulative species, until you finally reach sampling session number 10, with the cumulative species and new species caught numbers being the final values. With this, you're going to be creating a chart, a line chart to be specific. So to create the cumulative species graph, or chart in this case, we're going to need to first highlight our cumulative species segment, or column. After highlighting the column, you can go over to the Insert tab up here, right next to the Home tab, click it, and then glide your mouse all over here to the Insert Line Area Chart section. You can click on this little icon, the drop-down menu, and you can select either one of these formats here. I'm going to go with the first one, the line chart here. And as you can see, it has our label of the column, cumulative species, and it has our data represented as a graph, or an increasing line graph. This graph is what you'd use to interpret your data, or the data regarding your cumulative species analysis. This is where you're going to try and interpret why or how or what is happening with these graph points. Case in point, why there's an increase or sharp increase in number of species caught, or maybe why the number of species found, cumulative species found at one point, is 11 at this point. Essentially, you're going to be looking at all of the values here. You're going to be looking at how the curve changes, how it shifts, how it plateaus, how it grows, everything about the curve, and trying to decide or interpret or essentially pull together strings of ideas and concepts or reasonings to try and help you understand what's happening. And that concludes our Lab 8 exercise. Data analysis is really not that difficult of a task, albeit a bit tedious. But regardless, I hope this video helped you all to understand what we're looking for when it comes to Lab 8. If you have any questions, concerns, or anything, remember, raise it up with your lab demonstrators. Feel free to. We are here to help you out. And with that, thank you for watching, and I hope you all have a great day. Bye-bye!